that's where her doors are.
I mean, then we don't have to do it.
We'll see if we have extra time or not. I think it's kind of dumb to do the questions in online.
I've already learned about that.
I know.
Okay, so we've talked about this multiple times already, and oh yeah, so that's obviously the fire one. I hope they don't do this for a long time. No, and we're not even supposed to until later.
I can't. I had PE like 2 periods ago. What? 3 periods ago.
They have a schedule of some periods are supposed to do it now, some periods are supposed to do it at the end, some are supposed to do it tomorrow.
It should not go on.
Oh yeah.
Okay, so question number 1, very important question. Why does the cell have to copy its DNA before it divides? So, yeah, Leo, you want to go ahead?
So
so each new cell needs a full set of DNA, right? So we have to copy the DNA first so that when we split the copies apart, every cell can have an exact copy. And that happens in the S stage, in the synthesis stage, which is part of interphase, right? So we're going to pick up where we left off yesterday, which is looking at those mitosis stages. So if you have your folder open to that table, and I honestly don't remember how far I got. We just talked about the first 2 stages. Yeah, we're on metaphase. Okay, perfect. So towards the end of class, I was asking you to look at the differences between the prophase model here and the metaphase model here. And so you might remember from learning this material before that metaphase and middle both start with M. And so you'll notice at this point, all those chromosomes that packed up in prophase are now lined up in the middle of the cell, right? So and equally important, at this point, there is now a spindle fiber that connects each side of the chromosome to opposite ends of the cell. So spindle fibers. And again, I'm going to use a term from yesterday, sister chromatid. So each half of the chromosome there is a copy of itself, right? So because we've copied our DNA, and the term for that is a sister chromatid. So one sister chromatid's on this side of the central line, the other sister chromatid's on the other side. And that's metaphase. Pretty quick stage, really. So before we write down steps for the next stage, anaphase, talk to your group members. What's happening between this model of metaphase and this model of anaphase? All right. So, pretty obvious change here. The sister chromatids are separated, and they're going to be basically be pulled along those spindle fibers towards the opposite ends of the cell. So that's what we need to write something about that in here. So sister chromatids separate and are pulled along spindle fibers towards opposite ends. So one of the copies from each chromosome is going to go to one end. The other copy of each chromosome is going to go to the other end. Everybody's getting a copy.
You get a copy. You get a copy.
And once you write that down, take a look at the next picture with your group members. What's happening in telophase?
Yeah, they're probably pulling apart.
All right. There's actually a lot of things happening here in telophase, and in a lot of this phase is exactly the opposite of what happens in prophase. So if you look back at your prophase box, what's the first thing we wrote there? The chromosomes condense. So here the chromosomes start to unravel. So they unravel back into chromatin form. And then, did we talk about the nuclear membrane next then? Is that the next thing is prophase? So you'll notice around each one of these new nuclei, that nuclear membrane is starting to reform. Now I have two nuclei, right? The spindle fibers have done their job, so they're going to break down and disappear. That actually starts during anaphase, but they're mostly completely broken down at this point. And then lastly, I really probably should have put telophase/cytokinesis. Because you can see here the cell itself, the cytoplasm is actually starting to divide. Okay, so that is technically a different process, that's cytokinesis, but they kind of happen at the same time. Okay, so cytokinesis is the movement or splitting of the cytoplasm. So that's really exciting. And I'll have a couple questions in your worksheet today that will kind of distinguish a little bit more between mitosis and cytokinesis. But so, questions about any of that? Okay. So there's a picture down here with 4 cells. So there's one cell that's in prophase, one in metaphase, one in anaphase, and one in telophase. So go ahead and talk to your group members, see if you can agree which cell is in which stage.
Here. This one goes in what? Yeah. Right. No, metaphase is longer. No, this one. And then I think the top right is anaphase.
Yeah, I don't label these A, B, C, D up here. Okay. So which of those 4 cells do you think is in prophase? A, good, yeah. So in this case, we still have our nucleus, but we can start to see the chromosomes that are condensing, right? So we can see those condensed chromosomes there. So that's prophase. After prophase comes metaphase. Which cell do you think is in metaphase? C, good, yeah. So here all the chromosomes are lined up in the middle of the cell. That's metaphase. After metaphase comes anaphase. That is cell B. So we can see the chromatids have separated from each other. Half are going that way, one half going that way. One full set of DNA is heading towards each end of the cell. And then that would leave letter B to be telophase. So once those chromosomes reach the opposite end, all those reverse processes of prophase happen. So if you turn the page in your folder, or actually you don't have to turn it, well, I mean, it's on the next page. I moved the picture around a little bit so I could have them both at the same time. We're going to spend some time kind of filling in the questions in the next few pages. We do some kind of questions like this. We do this fairly often, but this is really a guided discussion to work through in your group. And most of these questions are going to relate directly to the model that they give you. So most of these questions are going to be focused on model 1. Some of it's on information we talked about yesterday. So I like to give you some time in your groups to do questions 1 through 9, so all the way up to 10 if you want to make a mark right before question 10 so you know to stop there. And I really do think that you understand material better once you're able to put the material into your own words. So I really do want you to talk through this as a group and not just do the work individually. Capiche? Okay, so go ahead and get started on that. Questions 1 through 9. Some of them are really easy. Yep.
Oh, dude.
I think you can probably do them in order.
Oh,
Because the prophase is just the product of prophase is the product of what the phase is.
no.
I don't have a razor. We're gonna answer. We're gonna be. That was me though. That was me. I got it. There's no day.
So meditation is all part of it. So this is what. So while prophase is part of my it happens before G1.
Okay.
So it's not the
Oh wait, I think she was a single the replicated ones are yeah, it's a single
that's called problem. What? Literally just draw a circle. Oh, yeah. What is the replicated? Oh,
Go back. The replicated one is this top.
Hey, how many replicas of the wall?
So this itself, this program is already copied.
There's two kinds of
So this is actually one of yours.
This is the same. This is
So if you're a program that
What happens when you
That's
Oh, wait,
right.
oh, oh, that's like 20, that's 20 on the one.
Separate. So this is what it
So there's two.
Yeah.
So then, okay, I got one. What if you-
So, yeah.
Yeah, we're here.
Okay, I can see that. Okay, now that's how many months is that? It is, but it's saying how many. I'm not 100%. I wrote, what? Okay. How many reps?
You, if you want to work ahead a little bit, you can, but you can stop before number 10.
Yeah, but count one per person. Yeah, so yeah, count two of them as one. Count one. That's fair. This is
I vote for EMA.
So,
I feel like I don't really know it really.
I got question 9 just before you said
That's okay.
yeah, but it's safe. It's safe about pass.
Oh.
Because it's saying use a company back for the truck. Did not occur before that. So before that. When you saw me, I think
Whoa, it's 2 parts.
Not fair. I mean,
I read it.
All right.
No, I would not fully continue to hire the cell phone. Well, is he?
Sure.
Also, every time you have support separate, anything. And yeah, not the day after. Not the day after.
It's good. Okay.
I don't know why.
Yeah.
They're waving out. I should have done that. Geometry stuff.
That's a great.
And then you're. Yeah, I tried to wait 5 months.
I don't know.
Now we're saying,
I put.
Alright, folks. We're gonna go ahead and go over these first 9 really quickly. If you didn't get all the way finished, that's fine. So hopefully these start out pretty easily here. So number 1, the 4 stages, they're just listed here at the top of the model, right? Prophase, metaphase, anaphase, telophase. If you look at the cell cycle, the M phase happens before G1 and after G2. These 3, G1S and G2, are all part of interphase. Before mitosis, we start with 1 cell. After mitosis, we end up having 2 cells. I think 5 is a little bit tricky, so I think it would have almost been better if they said draw a replicated chromosome first, because that's labeled for you in the diagram, right? So this is a replicated chromosome. It would probably be better if we drew it something like this. Okay. So this is replicated because it's already been copied, right? So this X shape contains 2 copies of the DNA, and we call those sister chromatids. So eventually, when we get to anaphase, one of those sister chromatids, 1 copy is going to go to 1 cell, the other copy is going to go to the other cell, right? So if you look at telophase, this is what a single chromosome looks like, right? Just 1/2 of that. So you can make it curved if you wanted to, but that's the single. Just 1 chromatid, just 1/2 of the replicated chromosome. So any questions on that? I think that was one of the harder questions. So number 6 then says, how many replicated chromosomes do we have here? We have 1, 2, 3, 4. I'm sorry, did you have a question? Oh. So we start with 4 chromosomes here. A copy of each of those 4 chromosomes ends up in each cell. So each of these new cells also ends up having 4 chromosomes. Okay, that's a little bit like, I do think that's a little bit confusing, but you just have to remember that when the sister chromatids are connected, that's still considered to be 1 chromosome. When they're separated, then they're now 2 chromosomes. So we have 4 replicated chromosomes here. We have 4 single chromosomes in each one of the new cells. You guys know how many chromosomes we have in every cell? Yeah. Oh, close. Well, I mean, so we actually have 46. So, yeah, 23 from mom and 23 from dad. And we'll talk more about that later, but this is way simplified. So we'd actually have 46 of these in the nucleus when we start, and then 46 individual chromosomes in each one of our new cell when it's done. Number 8, this was really from if you look back at our notes yesterday, a chromosome is just DNA that's wrapped and coiled up right around those histone proteins. So it's just condensed chromatin, condensed DNA. number 9, I heard somebody mention a checkpoint, so that's good. Normally this would be something we would catch in one of the checkpoints. But if we didn't replicate all of our chromosomes before we started this process, when we got to telophase, what would be different? Yeah, you'd only have half of the chromosomes. So if we didn't duplicate the DNA, we'd only have 2 out of the 4 chromosomes in every cell. We'd only have half of the DNA. And if they didn't divide equally, you might end up with like 3 in one cell and 1 in the other, right? So the DNA wouldn't get divided properly, ultimately. And if that happens, if we don't end up with a complete set of DNA, it's very likely that the cell is not going to be viable, right? So part B said, would the new cells be viable? The answer would be probably not. Because in most cases, we need the full set of DNA in order to be able to function. So we feel pretty good about that? Anything on that? Okay, so the next set of questions, 10 through 20, give you some time to work on those. We're probably not going to go over every single one of those questions as a class because we're going to run out of time. So if you do have a question on them, try to raise your hand. I'll come over and answer that with you. You can skip. If you take a look at number 17, you can skip that because we just did that table as a whole class. So you can skip number 17. But I do want to point out that in this table, they did actually make telophase different from cytokinesis, even though they kind of overlap. So just to keep in mind, telophase and cytokinesis are technically 2 different processes. But take some time to work on that with your group. Let me know if you have any questions as you go.